we're denaturing RNA or DNA before running a urea-PAGE gel, we don't just get it hot, we then get it cold. We stick it back on ice, and this might seem counterintuitive, but it's really important to prevent the reformation of those secondary structures and of the double-strandedness that you were trying to avoid. Let me explain. We can use gel electrophoresis to separate molecules based on their size. So this could be proteins, this could be nucleic acids like DNA or RNA. We send them traveling through this gel mesh, and the bigger ones are going to get slowed down more, and so they won't travel as fast. And when you turn off the gel, you turn off the power, you stop them moving through the gel, the bigger molecules are going to be farther up, and then when you stain the gel, you can see different bands corresponding to the different size molecules in there. But what do we really mean by size? Are we talking about like how many amino acids it has, how many nucleotides it has? Well, if we want that, then we need to make sure that its size corresponds to like its linear length. And if you have a linear molecule, then this doesn't matter. The length is going to be like the same. So if you have like double strand, just like a double strand of DNA, normal agarose gel, no issue. The length is going to correspond to how fast it runs. But what if you have something that's folded up? So if you have something like a protein that's folded up, or if you have RNA or single-stranded DNA, these can actually fold up upon themselves to form various shapes in the case of RNA. And the single-stranded RNA, well, if you want it to stay single-stranded, you're going to have to prevent it from, you're going to have to denature or pull apart any double strands. Because when you then run on the gel, if you want to run a single strand of DNA or you want to run a single strand of RNA, you want the size to correspond to that linear length rather than to its shape, because that shape is going to influence how it runs through the gel. And so when we run a protein, denaturing protein gel electrophoresis like SDS-PAGE, we use the detergent SDS to denature the protein before we run it. When we run a urea-PAGE gel for denaturing RNA or DNA gels, basically what we have here is that we use a denaturant called formamide in the loading dye, and then we use urea, another denaturant, in the gel. And basically what these denaturants are going to do is they're going to replace the bonds between those hydrogen bonding interactions between the different nucleotides that are keeping the RNA in that little pin, or that are keeping those double strands of DNA together. And they're going to kind of replace it. But they can't do it alone, because those bonds that are formed, those structures, those are going to be really stable. They're the happy place for these molecules. And so they're going to stay in their happy place, and they're not going to want to be disturbed by these little denaturant molecules. So instead, we have to give those denaturants help. We do this by using heat. So before we run the gel, after we load, we mix it with that sample loading buffer, which has that formamide. So it has one of those denaturants, and it also has a dye so we can visualize as the gel front moves through. It's not going to show us the RNA or DNA, but it will show us just the dye front, so we know when to turn off the gel. It's also going to have something heavy so that it sinks in the well. But actually the formamide is heavy, so you don't really have to add it in that case. But anyway, so you mix that with your sample, and then you heat it up. You heat it up, typically I do 90 seconds at 80 degrees Celsius. Some people do longer at a lower temperature, or shorter at a higher temperature. You don't want to go too high in the temperature. You don't want things to evaporate and then end up with less volume and end up with things sticking to the side of the tube, all that sort of stuff. But the heat, you need enough heat that it's actually going to allow those molecules to unfold. And once they're unfolded, now that denaturant can sneak in there. So make sure that you give the tubes a quick spin before you actually load your gel, so that anything that has evaporated will come down to the bottom of the tube, and then you make sure that you can load it all. So if you think about the folded up molecule or those double strands of DNA, there's a lot of interactions. It's not just a single base pair. You would have a whole string of them, and they're right next to each other. So if one of them comes apart, then it's snaps right back together because its neighbor is pulling it in. But if you think about those denaturant molecules, well, each of them, it can only form limited interactions with the molecule. And so if those fall off, well, now they're going to float away, and it's harder for them to come back. So this is why we can't just use the denaturants alone in order to try to break up these stronger interactions. Instead, we give the molecules heat. Now, what the heat does is it's going to allow the molecules to wiggle around, and when they wiggle around, it's going to be harder for them to stick together. You see, molecules, although we talk about them as kind of I say, they like to hang out with this, well, really, they'd rather be alone. They're loner. They'd rather be going on by themselves But these other molecules, they're just attracted to them. And they need enough energy to break free from those attractions if you want something to unfold or denature. And so we give them the energy in the form of the heat. And once we heat it up, well, now there's the opportunity for those denaturing molecules to sneak in there. And once those denaturing molecules are around, it's harder for these molecules, the RNA or the DNA, to find those binding partners that they were bound to before, because you have all these denaturants just kind of getting in the way. Now, when you heat it up, the denaturants can come in. But when you take away that heat, well, now those molecules are going to kind of like get stuck again. We want to make sure that they're not going to get stuck in those super stable interactions and then run through those stable interactions through the gel. That's what we want to avoid. So in order to do this, we can't let it slowly cool. If we slowly cool it, well, now it's going to kind of wander around and break free from those like looser interactions because it has enough energy, and then only when it meets those stronger interactions, when it makes those strong interactions, well, now it doesn't have enough energy because it's cooling down, but it still had enough energy to break off those weaker interactions, but not enough energy to break off of these stronger ones. And so they're going to get stuck in that structure that we tried to erase in the first place. So we don't want to cool them down slowly. We don't just want to stick it off of the heat and just stick it on in just like room temperature before we load our gel. Instead, we want to stick it on ice. What this is going to do is it's going to take away that energy really fast, and it's going to take it away so fast that those molecules aren't going to have time to find their like Prince Charming. Instead, those denaturant molecules, well, now they have a chance, because those molecules can't, when you lose the energy to a level where you can't break free from those denaturants, well, now there's not the opportunity to find that other partner, and you don't have the energy to go seek out your soulmates. So what's going to happen is you get these molecules that are going to stay unfolded. And then once they're in the gel, you get a little heat from the gel running. You get the urea in the gel that's going to take the place of any formamide that falls off. And so your molecules are going to stay unfolded. They're going to stay single-stranded, and all of this great things throughout your run. So this is why, in order to denature RNA or DNA before running a gel, we add this denaturing containing sample buffer. We heat it up, so for like 90 seconds at 80 degrees Celsius is what I do, and then stick it back on ice for about a minute and a half or so before you load your gel. This is going to make sure that those molecules are going to unfold, the strands are going to come apart, and they're going to stay apart. So this is why we do that quick cool, so that they can't find those happy soulmates. Instead, they lose their energy too quickly, and the only interactions that they can form are these weaker ones with like the denaturant and that sort of thing, which is going to allow us to then run them through the gel because they're not going to get trapped in a stable shape. And so hope that helps you understand the kind of like paradoxical seeming heat then cool, of running one of these gels.